Tonight we are delighted to have as our speaker Jeffery Schloss of the National Human Genome Research Institute of the National Institutes of Health. In 2004, the Institute launched an initiative, an innovative program to reduce the cost of DNA sequencing by 100-fold in 5 years, and ultimately to reduce those costs by 10,000-fold. This would bring the cost of whole genome sequencing down to $1,000. And at that level then, whole genome sequencing becomes part of an individualized medical care plan. Tonight, Dr. Schloss will tell us a little bit about where we stand today. He will outline how costs have been substantially reduced and how the new generation of sequencing technologies is making it possible for us to reach that $1,000 goal. He earned his BA in biology cum laude from Case Western Reserve University and a PhD from Carnegie Mellon University. He currently manages the grants program in DNA technology development at NIH, including the $1,000 Genome program. Jeff Schloss.
Thank you very much for the invitation and the pleasant evening we've had so far. And my apologies for the screensaver. I'm going to have to go to NCBI and download the one of the reference genomes, and I'll just have that streaming as my screensaver for future reference. So this is a very diverse audience, a little different audience than I usually speak with, and so we'll see how this goes. Okay, this is not, this is frozen. That's what it was. Okay, so what I'll try to do today, and I will watch my time so that we have time for questions and answers, is I'll tell you a little bit about how the program came to be, a little bit about how we started to implement it. I'll give you a little status report. I won't go into a lot of detail on details on how the technology works, but I'll give you some quick overviews, because I think you need to understand sort of how the basic technology works in order to appreciate how we've made the leaps forward technology-wise that are driving the costs down. Then I'll describe where we're trying to go, how we think we'll actually achieve the cost targets, and then talk briefly about why it matters and some context for why we're trying to develop technologies, and then wrap up with a little bit more about the implementation of the program, and perhaps some of the reasons why it's worked as well as it has. And the bottom line there is, I think, in large part, we were in the right place at the right time. A lot of things had gone before. So I'm going to start the story as it was becoming clear that we were breathing a sigh of relief. It was becoming clear that we actually were going to complete the sequence of the human genome. I should say that when the Genome project was started, it was a very, very bold plan. Nobody knew how we were going to get there. Technologically or conceptually. And so it wasn't clear until shortly before the goals had been accomplished that we would actually get there. So we were getting there, and we needed to say, needed to ask the question, what are we going to do next? The Genome program being largely in the United States a government-run, government-organized program, one possibility would be that the organizations that were running it would go away. That rarely happens, and we actually felt that there was a lot of important additional science to do. So we weren't quite ready to go away, and we talked about plans. So one thing that was very clear was that having achieved having determined the sequence of the human genome, we were not done sequencing. There were those at the time who said, we've sequenced the human genome. Now let's move on to something interesting like proteomics, studying all the proteins. But it was very clear that in order to understand that genome, we had a lot more to do, and its importance for health and disease. We had a lot more to do. One of the things to do was to sequence genomes of many other organisms. And without going into a lot of detail, we're all part of the unity of life, and the genes that you find in the human have their counterparts in the genes of most of the other animals, and in fact many plants on the earth. And so by sequencing genomes from organisms at various evolutionary distances from the human, you learn different kinds of information, either about protein coding regions or regulatory regions, and organization of the genome and so forth. So this was one of the reasons why we wanted to do a lot more sequencing. So that's captured up here in comparative genomics. But we'd only sequenced in any particular region of the genome one human. The way the Human Genome Project was done was we had a number of volunteers who gave blood samples, DNA was collected from them, and then we purposely actually generated sequence from a mosaic across those multiple individuals so that we would not be sequencing one individual. For the scientific study at that point, this was from an ethical, legal, and societal perspective, this was probably the right way to go. But ultimately, we really wanted to be able to get down to the genomes of individuals, particularly, first off, individuals who have or don't have particular diseases and conditions. That is, we wanted to study human genetic variation to understand the genomic contribution to disease. So there were a lot of different kinds of information. There was a lot of different kind of information we wanted to get from human genome variation. Clearly, although the focus from the National Human Genome Research Institute at the National Institutes of Health was on human genome sequencing, this technology, if we could develop it, would have importance for agriculturally important species, for understanding microbial communities, for medical reasons, for environmental reasons, because of the impact of microbes on our food supply. And it hadn't escaped our notice. There was a lot of interest in the use of sequencing and other DNA technologies for monitoring for bioterrorism events. And so one thing that I'll just say is that in our development of the program, and I'll come back to that at the end of the talk a little bit, we consulted with numerous other agencies of the federal government that had interest in these technologies for many different reasons, and have maintained contact with those entities throughout the program. So I just want to go through a couple of slides to describe to you the technology that we used to sequence the human genome for the Human Genome Project, mostly in order to lay out the contrast with the technologies that exist today and the ones that we're trying to develop. So if you, the human DNA exists in our chromosomes. There are 23 pairs of chromosomes or 46 chromosomes. Each of those is an individual, very large DNA molecule, on the order of 130 million bases within each of those molecules. And some are longer and some are shorter. So if you take that, consider that whole collection the genome, the way we had to do the sequencing, just because of the technology that we had available, was to shatter that DNA into pieces. And we did that in an organized way. First, we shattered it into large pieces, and then we shattered each of those large pieces into smaller pieces, and each of those into yet smaller pieces. We'd actually built maps as the first several years of the Human Genome Project in order to be able to figure out where we were in the genome. Because again, the technologies that we had available could only sequence short pieces of DNA. So we built these maps, and we were essentially going to hang the sequence off of those maps. The technology that was used for sequencing the genome, and again, I'm not going to go into any of the details here, but for each of these little fragments, and these little fragments of genomic DNA were actually cloned in bacteria so that we could make many, many copies of each of those original fragments just by growing the DNA in bacteria. And then isolating the DNA from the bacteria and running some biochemistry. The biochemistry we were using was developed by Fred Sanger, so we call it Sanger sequencing. And what it does is it generates nested fragments of DNA where one end is always the same and the other end is different distances from the starting end. And so you'd separate those, that nested ladder, if you will, on a technology called electrophoresis, where again you just display molecules based on their size. And in one set of reactions, the fragments that ended in G's were labeled. In another set of reactions, the fragments that ended in A were labeled. G, A, T, and C being the four nucleotide bases that comprise DNA, and so forth. So you'd run these reactions, you'd run them on gels, and essentially then you could read off. So the sequence here would be A, G, T, A, C, T, and so forth. And that was, initially that was done manually, just as I showed you, but starting in the 1980s, a machine was developed in which the fragments of DNA were labeled with fluorescent dyes that glowed under illumination, and with a laser shining at the electrophoretic gel, you could actually watch those different colored fragments passing down the gel and read off the sequence. And so this was in a later version of the instrument. This is what a series of lanes, in this case 45 lanes on one of these gels. This is the way the readout was reconstructed on the screen. And so the computer would read those fragments and you'd get peaks and call the peaks for the bases that they represented. So this was implemented on a number of different, on a series of generations of instruments. The last one is shown here. This was actually the instrument, or the type of instrument on which most of the Human Genome Project per se sequence was collected. And there were 96 different lanes, and you could run, this was a robotic device, so you could load samples onto the machine, and it would run for several hours or a day and read off those reactions. So this summarizes the workflow ahead of running the machine, and I've actually mentioned most of these steps, but that is every fragment of DNA had to be cloned in bacteria, grown up so that you'd have enough DNA to work with. There were robots that picked those little bacterial colonies off of a plate, put them into reaction tubes, or microtiter plates in this case, and then large robots that ran all of these reactions, set up all the biochemistry, ran all these reactions, and then these were loaded on the sequencing machines, 100 per run. And the best that these machines got was about a 2-hour run to collect the data, and then you'd load the next batch of samples. So you could run 10 or 12 cycles of loads, each of those of 100 lanes in a day on one machine. And the factories where this was done, often had 100 of these machines. They were really run as factories. They were set up in academic labs, but these people had gained a lot of expertise over the years, and were running much more in the style of factories. During the time that the Human Genome Project was underway, we were tracking costs, and this graph shows what the costs were per finished base of DNA. And through a series of technology improvements and automation and economies of scale, by the time we completed the human genome sequence, which was estimated to have cost about $650 million for the sequencing part per se. You see the number, $3 billion. That was the whole genome project. We were, without going into details, we were doing a lot of stuff. We were developing technologies, we were building maps, we were sequencing other organisms for practice and for to understand the content biologically of those organisms' DNA, the genomes, in order to compare it to the human. But the sequencing of the human genome per se in the Human Genome Project in the public effort was estimated to cost about $650 million. If you had done it again at the end with the technology as it had evolved, it's estimated that the cost would have been about $50 million to sequence the human genome. So over this period, we noticed it was taking about 10 years to reduce the cost by 2 orders of magnitude. And so as we were doing this planning process to say what are we going to do after the completion of the human genome, This was an article, the strategic plan published in Nature in April of 2000. We laid out a plan that would study genome, the foundation of the Human Genome Project, study the genomics to understand biology, and eventually move toward health and society. We had a number of cross-cutting elements. These were training of students, computational biology, technology development, ethical, legal, and social implications studies. And one of those was technology development, and we framed in this article a series of quantum leaps, which we described at that time as technological leaps that seemed so far off. These were actually, in another part of the paragraph, these were framed as dreams. Seemed so far off to be almost fictional, but if they could be achieved, would revolutionize biomedical research and clinical practice. One of these was genome sequencing of a mammalian genome for $1,000 or less. Out of that grew a grants program. We published a request for applications, and we said, in the RFA, we said current technologies in about 2004 can produce a sequence of a mammalian-sized genome of the desired quality. This was a high-quality draft, more on that in a minute, for somewhere in the range of $10 to $50 million. And the goal of this initiative, as you heard from the introduction, was to reduce this by 2 orders of magnitude and subsequently another 2 orders of magnitude. We made initial awards in 2004, and again, our goal was to within 5 years, so instead of 10 years to reduce costs by 2 orders of magnitude, we're going to try to cut that timeline in half, reduce costs by 100-fold in 5 years by 2009, and to about $100,000, and to about $1,000 by 2014. A cost goal is completely meaningless, of course, without some sort of a quality goal. And our quality metric for the technology development program, though it's certainly not good enough for medical use of sequencing, but as a bold plan for technology development, our quality goal was a draft genome sequence of the mouse. It was published in Nature in 2002, because there were a lot of statistics about the quality of that draft sequence, so we could actually have a quality measure. So over the years, as we started the program, we had a strategy of funding a lot of different technology approaches. Some of these were relatively conservative. They were miniaturization and integration of the process that had been used to sequence the human genome the first time. And then others were more forward-looking, more challenging technically in an engineering perspective. And for some of them, we really didn't even know all of the science that we needed to know in order to do the engineering. There was science that needed to be learned first. And so I'll be talking about some of those here. The big breakthroughs came within just a very few years within a short time, I should say, after we launched the program. And the method, instead of being called Sanger sequencing and then separation by electrophoresis, or capillary array electrophoresis as it was ultimately implemented, this new set of technologies is called sequencing by synthesis or sequencing by ligation, minor variants on each other. There were some of the molecules, some of the methods were single molecule sequencing. Most of them were ensemble sequencing, and I'll explain what I mean by that in a moment. And essentially all of them used fluorescence detection, which was the same way we detected the molecules for the Human Genome Project. So the first innovation here was that instead of having to grow up to clone all the little tiny fragments of human DNA in bacteria and grow them up and handle them each individually in a test tube, there was a way was developed to essentially build that library in one test tube for the whole human genome. And this graphic illustrates that. So this is one of many, many, many fragments of human DNA. And it was modified by putting short little pieces of DNA whose sequence was known onto the end of every one of those fragments, using one of those ends to capture that fragment of DNA on a bead, and then putting, now of course there are hundreds of thousands or millions of these beads in this test tube, and each of them was captured in a water droplet in oil. So essentially you build an emulsion like salad dressing, where you have little droplets of aqueous solution in oil. Each of those little droplets becomes a reaction tube. But now it's all in one vessel. But you have millions of different reaction tubes without having millions of different test tubes. And you amplify the DNA that was attached to the beads. So now, where you started with one genomic fragment from a particular region of the genome that was attached to a bead, that's amplified by a biochemical process. And all of the copies of the DNA on that bead arose from that individual fragment. So that they're all identical copies on any individual bead. And now you break the emulsion and collect those beads. And now that's your substrate for doing the sequencing. The way the sequencing worked is illustrated in this slide. So we start with this fragment of DNA that's on the bead. Add to that another short fragment of DNA called the primer that helps you prime the reaction to extend the DNA molecule. Add DNA polymerase, and add the reagents needed, all the other reagents needed to extend the DNA fragment. In this case, it's the individual nucleotides that are, and each one has a different color label. So all the C's are labeled, for example, red, and all the T's are labeled green, and so forth. So let the reaction begin, incorporate the first base. Now there's a blocking group on each of these bases, so that only one base can be attached at a time. Ordinarily in the cell, there's no blocking group, so DNA polymerase happens and you extend a long DNA molecule. But in this case, there's a little blocking group on each of these, so you can only add one base at a time. Wash away everything else, detect the base that was added by shining a laser on it. The fluorescently labeled fluor on the base is detected. It glows and it's detected. And then repeat this step one after the other. In the process of removing the blocking group, you also remove the fluorescence from this added T. So now you're starting as if you just had the cycle all over, and you add the next base, detect it, add the next series of bases, each time detecting, washing away the blocking group, washing away the fluor, so that you can collect the information. But remember I said we had lots of beads in the tube, so what you're doing is you're doing this experiment on all those different beads that are spread out on the last slide, all at the same time. And you're watching each bead, and you're watching the DNA grow, essentially, watching the addition of single bases on each of those molecules. And that's what the experiment looks like. This is one cycle. In the next cycle, the color on any of these positions might change as a different base is incorporated. So again, we've gone from a workflow where we had to handle every single DNA fragment in an individual tube with large robots, to making that library in a single small test tube, and then capturing the information on millions or hundreds of millions of different DNA molecules in a single write. So the throughput goes way up. This shows the initial commercialized versions of the machines that would do this. And as you see, they started coming out in 2005, and after that, there was a new commercialization of a new machine each year. The annotation here is a little asterisk. These are systems that received, companies or entities that received support from the National Human Genome Research Institute. We supported these at different stages in their development. So 454 basically already had a commercial machine in the works, and a grant from NHGRI helped them to scale up more rapidly. On the other hand, Agincourt is a small company that spun out of a university, and we supported them early on in the development. Ultimately, they were bought out by Applied Biosystems for the commercialization. So these are the current versions of these machines. The throughput has increased tremendously. Costs have come clearly way down. That's probably all I need to say about that. In fact, the throughput of most of these machines is so great that many labs can't handle the data. So these companies have come out with smaller versions of the machines for different settings. And also, actually, these are more likely to get into clinical settings more rapidly. This slide is an attempt to collect from a combination of company websites and from some practical, tempered with some practical experience, some characteristics of each of these initial machines. And without going through this in any detail, all I want you to glean from this slide is that the machines produce different kinds of data. For example, the length of the read varies from about 30 bases per read to 900 bases per read. Some of these machines, you actually, in order to run the full cycle, takes less than a day. Some of them take 2 weeks, and then different throughputs and so forth. This offers a number of things. One thing that it offers is competition. And part of the reason that the costs have come down so fast, and the throughputs have gone up so fast, is because there's been competition in this field. If there was one clear winner at the very beginning, we wouldn't be as far as we are. And this was one of the goals, actually, of the NHGRI program, was to try to facilitate there being more competition. Because at the time the Human Genome Project was done, there were 2 commercial machines on the market, but almost all the work was done on one of those. The other factor to keep in mind here is that with these different characteristics, there are actually different kinds of sequencing problems that you want to address, and they require somewhat different kinds of experiments. And so if you think of this as a set of tools, these different machines as a set of tools, you'd have a different tool that might allow you to do a particular experiment better, and yet when you wanted to do a different experiment, you might take a different tool. I won't extend that analogy too far. There are lots of different applications for these machines. Basic genome resequencing or de novo sequencing, and actually, I guess I won't take the time to go into the detail. We can look at variation amongst different individuals, and variation comes in different flavors. There can be local variations where one particular nucleotide is different from one individual to the next, but there can be larger scale variations too, where a particular region of the genome that's present in one person at a particular location is actually moved to a different location in another individual. And that has consequences for the way the genome functions. And I think I'll stop there. Actually, with these, except to mention methylation, we all think, we hear you have your DNA sequence and it's the same from the time you're born until you die in most of your cells. And that's probably true. But in addition to the DNA sequence, there are chemical changes on particular nucleotides within the DNA. And these can change throughout your life. And they're affected by environmental effects and developmental changes and so forth. And so while your DNA sequence per se, to a first approximation, doesn't change, these markings on the DNA can change, and they can affect how the DNA is expressed. And there are ways to using biochemistry today to determine that methylation status, and you'll see where we're going is technologies that can directly read that information from the genome. In addition to the DNA, as you probably remember and have read, the DNA is expressed in the cells through a process that produces RNA, ribonucleic acids, and it's from the RNA. So there are RNAs that are structural within the cell, and there are other RNAs that code for proteins. And so you can use these same set of tools, technologies, to read various kinds of RNA information. And the way those kinds of experiments have been done, and in fact some single nucleotide polymorphism or SNP determinations had been done, was largely using DNA arrays, which is not actually sequencing, but it's sort of a surrogate for sequencing, where you put lots of small DNA molecules down on a chip, and you let the DNA in your genome interact with that, and where there's a similarity, the DNA from the genome binds to the chip. Where there's no similarity, nothing binds in that position. And it's a different way of storing DNA. Similarly, you could do RNA expression analysis, in which cells is a particular gene being expressed or not being expressed. Could be done on those arrays. But the data you got out of those arrays was analog. You were always comparing two different conditions. So you'd be comparing intensity of a green fluor, green emission of the fluor versus a red emission of the fluor. So you're always reading off analog information, because whereas in these technologies, because of every bead has DNA that comes from a different region of the genome, you're actually doing a count. You actually end up doing a counting function. So you get a digital readout. So if you have something that's rare, some RNAs are expressed at very high levels, other RNAs are expressed at very low levels in cells. And so if you have those rare RNAs, they're really hard to detect with an analog signal. But when you can count them, you can determine quantification much more easily. So these tools not only allow you to sequence faster and cheaper, they allow you to do different kinds of experiments much more accurately than you could do them with the old technologies. I should also say that as these technologies were being developed, they were being applied immediately to many, many different kinds of studies. And so if you will, we had the push to develop the technologies, but we also had applications pulling the technologies in ways that would help the technologies to develop in ways that were responsive to the needs of the users. And this was really important. Again, I'm not going to go through these in detail. The point I want to make is that there were many different projects producing large volumes of data that immediately started to use these technologies and dramatically influenced how they developed, so the quality of information that they produced would be responsive to the users. And at the same time, producing useful data to perform these biological studies. So we had studies trying to understand the function of all the different elements of the genome. We studied individuals from around the globe. These were nominally healthy individuals to get a picture of the extent of human DNA variation in different geographic regions. We're in the process of sequencing 10,000 different animals to really nail down more of this comparative genomics. We have the International Human Microbiome Program studying all the different microbes that live in and on our bodies. You've probably heard this figure. There are 10 bacteria for every one of your human cells that make up our bodies. The bacteria aren't only harmful, we actually couldn't live without them. And so we have studies to catalog and understand the function of those microbes. And the International Cancer Genome Consortium, which is studying collections of large numbers of tumor normal pairs from many individuals for quite a large collection of different cancers. Again, trying to understand the genetic contribution to these cancers, what's going on with these cells. So what's the sort of the throughput, the data producing capability of these different technologies? So again, at the end of the Human Genome Project, using that technology in about 2003, if you had sequenced the human genome again at that time, it would have taken about 3 months with 100 machines, and you could have sequenced the genome. With these new technologies, in their relatively early days, in about 2007, you could do these calculations in about the same time, 3 to 4 months, but with one machine instead of 100. Fast forward a couple of years, and now down to 1 month from 3, less than 1 month with one machine. And today, it takes about a week to sequence a genome on one machine. But in fact, there's so much real estate, there's so much space on the slides, that in that week, you can get 3 genomes on that slide. And so we can sequence about, actually, 3 genomes in a week. You can't cut it short, it's not a third of a week, because it still takes a week to do the run. But three genomes in a week with one machine. And you've probably seen this kind of a figure if you follow the Human Genome Project at all. It is, here's 2003, so the first slide I showed you with costs ended about here. And so costs continued to come down, but at about the same rate until the introduction of these new machines. And that happens obviously right here. And as you see, a dramatic decrease in costs. And actually by 2009, the cost of sequencing a genome had fallen below $100,000. So we more than achieved that initial 5-year goal. The other thing you can see here is that this curve traces Moore's law, the approximate doubling of components on a computer chip every 18 months or so. It was an observation by Gordon Moore. It's not a law. It was an observation that this was how the computer chip industry was moving. And so we had the luxury that as we were sequencing the first human genome and proceeding beyond that, the computers were pretty much keeping up. The computer speed was keeping up with the data we were producing. But that's no longer true, and this is what we've run into a bit of a problem there. We're running into computer storage and computing capacity problems now, because we can produce genomes data so fast. So I want to tell you a little bit about some of the newer entries into the field and where they're going to take us. So this is a new technology that all the other technologies I've told you about use fluorescence detection. So they use cameras to record the data. Instead of a camera here, this company's technology uses a pH meter. And so I have shown L here, whoops, but this chip, and here's one of them, and I'll pass it around, has 1.5 million pH meters on it. Okay, so they like to say that they've got the most expensive, well, either the most expensive or the cheapest pH meter, because there are 1.5 million pH meters on that chip. So actually that should pass around the other one, because now the second generation of the chip has 7.2 million pH meters on it. And they've increased the size of the chip a little bit. There's a flow, there you see two little plastic rings. That's the flow cell in which you pipette your DNA, and then it flows through across the pH meters. So instead of using fluorescence detection, you're actually measuring the evolution of hydrogen, the release of hydrogen ions as each base is added. And that's just part of biochemistry. It's just how it works. The machine itself is small. It fits easily on this tabletop. I'm not advertising for the company. I'm simply stating some facts here. Whereas most of those other machines that I showed you cost on the order of $500,000, $700,000, this machine costs $50,000. And so those of you who are familiar with laboratory stuff, this is a 50 ml conical tube, just for sort of size scale. The tube is about this big. So this is a little tabletop device. Now, this doesn't have anywhere near the throughput. It doesn't have the read length yet. They're relatively early in the development of the system. But the idea is that they're using computer chip technology, and they ought to be able to scale using the infrastructure of the computer industry to scale these devices. There's a lot of work to make that happen, but that's the idea here. Another really intriguing new entry into the field is based on the idea of, well, what if we could watch single DNA polymerase molecules? Polymerase is the molecule, is a protein enzyme that replicates DNA in cells. What if we could watch those individual molecules in real time as they synthesize a new DNA molecule? Well, one of the challenges to doing that is that you need lots of nucleotides to drive the reaction. The nucleotides, again, are the building blocks of DNA. And now we're going back to fluorescence detection. If you're going to do fluorescence detection and you have a lot of fluorescent nucleotides right around the area where those new nucleotides can be incorporated, you have to have a way to filter out the light that these nucleotides are emitting and sort it out from the nucleotide that's at the active site of the molecule. And so I'm going to describe now two different approaches to doing that. One company has developed this device that they call the zero-mode waveguide. when you shine a light through a small enough hole, the light can't go through the hole. When you think about this, the front door of your microwave oven is designed to not let the microwaves out, because the little holes in the screen are too small to let the microwaves come out and blast you as you're standing in front waiting for your coffee to this is the same principle. You shine a light from down here, and if you have a small enough hole, the light doesn't go through, but you get a little energy field right there down at the bottom of the well. And so what they're going to do is they're going to pin their polymerase molecule at the bottom of the well, and there will only be enough energy to excite the fluorescent molecules right down there at the bottom. All the other fluorescently labeled molecules up here won't be excited, and so they won't give off light. And so that's illustrated here, and this is just a trace from one of their early experiments showing that, in principle, that it works. And now, this is this methylation thing. Not only can they distinguish A from G from T from C, but based on delays in how long it takes the polymerase to add the next nucleotide, they can distinguish a methylated A from a normal A. And so these are these epigenetic marks. And instead of having to do a big fancy test tube biochemistry and so forth, which is what we have to do today in order to detect methylated DNA sequences, they can read it off directly from the DNA molecule with this technology. So if they can make this work for all of the kinds of DNA methylations that we find commonly in genomic DNA, then this would be a very powerful tool. And in fact, I just heard a talk from one of the founders of the company this past week, and they've nailed 5 or 6 or 7 different common methylations that they can detect and distinguish from each other. So this is actually a movie. This is not a cartoon. This is actually a movie taken through the microscope of one of these chips. The chip has 80,000 zero-mode waveguides on it. And you can see them sparkling as each of those wells has a DNA polymerase and a DNA molecule in it. And this is real time. You're seeing DNA growing and recording it. And the way the zero-mode waveguides are on a little, I guess it looks a little bit like a little computer chip. And each of them is less than a millimeter in diameter. And then when they manufacture them, they just load them in these little plastic strips. And then these go into the robot. So this one doesn't have zero-mode waveguides in it. It's just for demonstration purposes. But that's how you would handle these things, to load them into the robot to do the experiments. So another characteristic is most of these technologies that I've talked about, if you noticed on one of those charts, they give relatively short reads. If you're trying to put those short reads back together, the way you basically do it is look for little overlaps from one read to the other, and then build them back up together. If you have a lot of, hundreds of thousands or millions of short reads, that's really hard to do. If you have longer reads, it becomes much easier to do. So this is the point of this slide, is just to remind me to say that this technology has given the longest reads ever achieved in any DNA sequencing technology. They see not very many, but occasionally a few of the reads are tens of thousands of bases long. So this would be very important too. I'm not going to go through this in detail. I'll just say that there are other ways to solve this problem of the high background of fluorescently labeled molecules. And so just to save time, I'm not going to go into the details of how to do that. And NHGRI has supported, and companies have tried to develop some of these other approaches. I want to tell you now about sort of a vision into the future of sort of what we think may be, and we're hoping may be one of the next technologies that will really drive down the cost. So I should say that with the technologies that I've described to date, the large commercial machines, costs have come down to about $4,000 or $5,000 to sequence a genome. That's if you're doing it, all the time, high throughput. In order to get that price, you have to not buy one genome. You have to buy 100 genomes. But if you're doing studies, you need to do that anyway. So you buy in bulk, right? But we're getting closer and closer to that $1,000 goal. But the qualities of the genome sequence that you get from those are good, but not exactly what we need yet for clinical application, and even for some of the disease studies we'd like to do. So this nanopore sequencing is something that we've invested in pretty heavily. That was originally conceived and published on in the early 1990s. This was the one where we said, if this is going to work, there's a lot of science we still need to learn. And so let me describe how this works. The idea is that if you had a channel, a small channel, that was about the diameter of a DNA molecule, the channel in this case is a protein channel that we normally find in the membranes of cells. And the function of that protein channel is to transport ions of potassium or sodium or calcium. And the concept here was, so if this would ordinarily transport ions, and we could make a measurement of how many ions were flowing across the membrane through the channel per unit time, would it be possible to put a DNA molecule through there? And would each of the nucleotides of the DNA molecule interrupt the flow of ions in a way where you could distinguish it among the 4 nucleotide bases? So that's the way the experiment would look, and people tried this with short pieces of DNA or RNA. And you could record signals, you could record interruptions in the ion flow, and those signals had dimensions. They had width and they had height in terms of the conductance change, how much ion flow interruption you could get. The dream was this, that is, you could clearly distinguish between the signals from A, C, G, and T. So this was conceived of, and people thought fairly soon after the concept, well, this is really neat, but if it's ever going to work, you'll never build robust devices that you can sell to people and they can use in their laboratories made out of proteins and lipid bilayers. All much too fragile, and you'll never be able to sell that. So we should make these out of hard materials. Well, the problem, and so why would we want to do this? First of all, if this works, you should be able to sequence genomic DNA directly. You would eliminate that last bit of the flow of the preparation of material, making those libraries on all the beads. You should eliminate all those steps and just purify genomic DNA and put it through the device. So no conversions, amplifications, essentially no reagent costs. You ought to be able to get very long reads, and I briefly described why you want long reads. You should be able to sequence the genomes of all the microbes that live in and on us relatively easily, because those are much smaller than our chromosomes. You actually could get back the material after sequencing. You wouldn't, because you don't destroy the sample. As I've mentioned, you should be able to sequence A, G, C, and T and those modified bases. You probably should be able to sequence RNA also. It would be digital. It should be very fast. And it should be fully electronic. So you wouldn't need cameras, you wouldn't need light. If all of those things are true, you ought to be able to make portable handheld devices, which would mean that you could use them. That's a tricorder, by the way. You could deploy them in. I thought I had one more program here. You could deploy these in resource-poor environments and potentially in doctor's office settings, and not have to do this in large sequencing labs. So, and as I mentioned, you would simplify the workflow. So people did start working on this. As I said, they thought that you would have to make these out of hard devices, out of solid materials. Part of the challenge here was that we did not know how to make these devices. Nanotechnology had not advanced to the stage of being able to drill pores of the dimensions that you needed into devices. And so that had to be developed. And a lot of that fabrication development work was not supported by NIH, but was supported by other agencies, such as DARPA and NSF. And part of the motivation for using that technology in this way was to solve this problem. So I'm just, in these two slides, I'm not going to go through these in any detail. I'm just going to say that there are a number of different device concepts. There are other ways to detect the DNA in the pore instead of interrupting the flow of ions through the pore in this dimension. There's a concept of being able to tunnel electrons across an electrode and see how the DNA changes that tunneling current across the electrode. That's hard to do physically, though it was demonstrated by computer modeling that if you could do it physically, you could probably distinguish amongst the bases. Recently, there's been a lot of attention paid to this relatively new material called graphene that may help to solve the problem of producing very tightly controlled, atomically controlled nanopores of appropriate dimensions, both in their diameter and in their height, and allowing you also to hook up your electronics directly to the material that is your nanopore. And experiments have been done to show that you can get DNA through devices like that. You can measure signals, but so far not close to sequencing. I'm going to skip this. There are some other ways to do the tunneling readouts, but I'm running short on time, so I'm not going to describe it. I want to wrap up this section of the talk by describing the rather significant advances that have been made coming back to this use of protein pores. First of all, people have shown that these devices made of protein pores, inletted bilayers, are really quite robust. They're not as fragile as people thought. And within the last 2 to 3 years, really stunning results toward sequencing DNA. We're not there yet, but results like this are why people are getting so excited. This is an experiment in which 2 different sort of styles of modification were made to the protein pore, both amino acid changes were made in the protein structure, and then chemical modifications in addition to those amino acid changes, so that you could attach various things to the protein pore to improve the readout capability. And this experiment shows the first time that people could distinguish among A, C, T, and G. In this case, a solution of nucleotides. So it's not a DNA molecule, but it's a solution of the building blocks of DNA flowed through this modified alpha hemolysin protein pore. And you could get 4 different signals, one for each A, C, G, and T. And in fact, not only 4, but 5. You could distinguish methylated C from regular C. So again, this idea of directly reading out methylated bases. And I'm not going to describe one way that you could actually use that kind of a device. There is a way that you could actually use that capability for sequencing. But I'm going to jump forward to where we would really like to be, which we call strand sequencing. And actually, is putting intact DNA molecules through. And so this next series of slides, which again, I'm not going to go through how the data are generated, but I want to give you a sense that people have done experiments that show that you can, in an intact DNA molecule, distinguish C from T from A from G, and resolve individual bases within a DNA strand by using alpha hemolysin. And once again, strand sequencing that not only distinguishes A from C from G from T, but 2 different methylated Cs. People have developed different protein pores that have better characteristics than the alpha hemolysin, which you see here has a long barrel. And in fact, there are about 10 DNA nucleotides in there, which makes it harder to read. But here you see a more funnel-shaped pore, which should make it easier to read. And in fact, you get very strong signals distinguishing A from Z, C from T from G. And here's another kind of experiment to show. So in this case, we're not distinguishing A from C from G from T, but instead we're stepping the molecule under tight control, 1 base at a time, through the pore, through the reader. And that's the other thing you need to be able to do, is you need to be able to control how fast that DNA molecule goes through. So we had single base resolution for reading, and here this experiment shows single base resolution for moving, for translocation, for moving the DNA through. And this is another way to show that. Furthermore, there are other ways to control the speed of the DNA molecule going through by modifying the protein or actually by building a device, once again, out of hard materials that has very thin layers. These are layers of metal or insulator, and we can call it a DNA transistor, that are the thickness of those layers is the same dimension as a single nucleotide of DNA. And then by using electrophoretic force to drive the DNA molecule through and switching the bias on that transistor, you would be able to ratchet the DNA molecule through the pore one base at a time. And this, I'm going to skip this just for time. An additional way to get control. So what I want to leave you with the impression in this part of the talk is that we have all the pieces. We can read, we can step the DNA molecule through, and now people are trying to bring those pieces together. And I'm told by multiple different people who are working in this field, many of whom are competing with each other, that within the next year we should see sequencing of DNA. It's not going to be a commercial machine. It's going to be a laboratory demonstration of sequencing of DNA with a nanopore. Why do we care? Because you have to slow down the transit of the DNA molecules through these sensors to about 1 millisecond per base in order to take a reading, just to get a signal to noise. So you have to slow it down. You slow it down to that extent. And you have about 1,000 nanopores in an array. And if you can build one of these things, then you can easily have an array of 1,000 nanopores. It would take well under a day, and you'd have these very long reads. You ought to be able to assemble your genomes. So we're getting to the point where sequencing the genome is not actually the biggest bottleneck. There are other bottlenecks. Some of them are in preparing the DNA to go into the sequencing. As I mentioned, storing the raw data. We're producing so much data now. The NCBI, which stores all the sequence information for public access, has actually stopped storing, has told the community they're going to stop storing certain sets of data because it just takes too much storage. These are potentially valuable data sets. These are new technologies. People want to be able to go back and look at the raw data because we keep improving the base calling software, but they're having to stop calling the bases and putting it all assembled, putting it all together, analyzing it for what's the difference between this individual's genome and that individual's genome is a big challenge. In fact, it's hard to get enough samples to do the disease studies that we want to do for various reasons, many of which are related to human subjects protection. So we have all kinds of other bottlenecks. This is just to represent the computational bottleneck, storage of information, software to analyze it, and the people to do all the work are bottlenecks at this point. Where would we like to go? We'd like to go to the point where, I mean, $1,000 is still too expensive for many things that we would like to do with genome sequencing. If we're going to project forward, it's not at all unreasonable to be able to collect genome sequence information in an hour or so, or less than an hour, for $50. The challenge is to get good enough quality information for whatever we need for the application. We don't yet know what kind of genome sequence information we need for individuals. But whatever it is, we've got to get a quality and scope sufficient for individual medical decision making if we're going to use it that way. Whatever that wet process is or electronic process has to be completely integrated with the computational tools and with electronic medical records if this is going to be useful. Here's where deployment in wherever you want to, basically, without having to have large clinical labs or whatever. The applications are for infectious disease, microbiome research, pharmacogenomics. This is drug reactions. Encoded in your genome are enzymes that can determine whether a particular drug will be effective for you or not, whether you'll get an adverse drug reaction, whether you say, okay, this person can't metabolize this drug, there's no point in giving it to them. Let's skip over this drug and give them another drug, which they can metabolize and may work for them. That's all I want to say there. So the program runs by, obviously, we issue requests for application. it's an essential component of this program that's made it successful is we have an annual grantee meeting every year. And these grantees are sharing information. I have a slide to make that point. Let me skip over that comment for the moment. The grantees have come together and published papers together. And they've published many research results, and we've recorded all that on our website so that it's easily accessible. So why does this work? As I said, the grantees are sharing information. There's a lot of information that's sort of secret to the success of each individual project, but there's a huge amount of information that they can share with each other at these meetings, and that really raises all boats. It's really accelerated the progress of the program. People are really motivated when they come to a meeting every year and find out how fast the competition is moving. It excites the students. They work harder. The PIs work harder. So this has helped keep the field moving. People are competing and collaborating almost at the same time. Many collaborations have come out of this program, in part stimulated by the meetings we've held. This is a great opportunity for students. They can move from one lab to another, gain experiences, they get hired by the companies. We've added an open day of meeting to our grantee meeting, so anyone in the community can come. We have people from companies, from the press, people who would like to get into the field coming to these open meetings, so we've helped to disseminate the information. Another set of essential components for making a program like this work. Our institute is really committed. Almost everything we're doing now in terms of the understanding of biology and the relationship of human polymorphism to disease is based on being able to collect information with these kinds of technologies. It's been really important. So the institute is committed. There's a level of trust and integrity amongst the staff members, so people trust each other and believe each other. We say when we need an investment in this area of the research, people go along with it, even when resources are constrained. We run the peer review of these applications within the institute. NIH likes to tout the fact that peer review is the foundation of the NIH way, and the NIH peer review system is quite good, but it tends to be rather risk averse. And to run a program like this, we have to be able to fund risky projects, and so we have to be able to run the review, to constitute the review panel, and instruct the review panel in a way that will allow us to fund the projects that need to be funded, that are risky to make the leaps forward. We have an advisory structure that is also willing to accept that risk, and where we can propose creative solutions for funding, and our advisors go along with that. And then, perhaps most importantly, there's a chain of discovery and development and investment out there in the rest of the community. So people recognize that there's a market, there's a research market for these instruments, and there's anticipated to be quite a huge clinical market for this kind of instrumentation and for the information that it can produce. So again, we're doing this ultimately. I don't know if you can make that out. These are all faces of people, and we're trying to move this as rapidly as we can to the clinic. This is not simply an exercise in studying biology. And over the last year or so, there have been increasing numbers of publications where the application of whole genome sequencing to particular clinical situations has either revealed new possibilities for newborn screening. It has guided therapies only in a couple of cases so far, choosing between two potential cancer therapies where there's an established practice in the field, and they couldn't distinguish using the established tests in the field which therapy should be used. Through whole genome sequencing, they were able to distinguish and give the patient the appropriate therapy, and the patient is at least in remission, and so forth. So there are examples of bringing this technology into real clinical use. Realizing that was happening, we developed a new strategic plan recently, and it's based in very large part on these new technologies that have been developed since that last strategic plan. And what we're trying to do now is move our investment, which was originally in genome structure, and over the past several years has been in understanding the structure and biology of genomes. And then more and more we're trying to move over here toward the right, if you will, toward effectiveness of healthcare, bringing genomics to the effectiveness of healthcare. So I'm going to let the last thing I want to show you is just, I needed to show you the rest of the information in order to tell you a little bit about where we were at the very beginning that enabled this program. So I mentioned we started back in 1995, and NHGRI funded the first grant to Dave Deemer, who was developing these protein nanopores for sequencing. In '97, we funded this investigator at Caltech named Steve Quigg. It was a new investigator grant. He was proposing single molecule detection, single nucleotide addition by polymerase, photo bleach to prepare for the next cycle. It's what I described to you about how sequencing by synthesis works. This technology became the foundation for one of the companies. We funded, so Illumina is one of the companies whose instrument I showed you. We did not fund the development of their sequencing instrument. We funded the development of their previous technology in microarrays, and this was in February '99, decoding of massively parallel hybridization arrays. A lot of that basic technology goes into the sequencing technology. And here are these two sequencing by watching DNA polymerase molecules in real time. We funded early small business grants to these two groups, 2001 and 2002. So these were all before the program formally started. We were working in this area, but didn't have a large focused program. In a planning meeting that we held in December 2001, this was in preparation for the 2003 strategic plan. We had a set of meetings, and this was the first time that I'm aware that the phrase $1,000 genome was used. We were trying to imagine the future. In July of the next year, we had the plan for this program. There were 25 academics, and they were biologists, chemists, physicists, engineers, people from sequencing centers, people with a track record of innovation and invention, and of handing off technologies they developed in their academic labs to companies. Those are the kinds of people we invited to this meeting. There were 10 different genomics companies represented. There were 5 other agencies of the US government and 2 other institutes of the NIH. And then so, as you see, there were additional investments. Synthetic nanopores funded for the first time in 2002. We met with 454, which was the first company that commercialized one of these instruments, in September of 2002. This is when they first, so they had been working on this for a while. These developments were going on, investments were being made, and they had their commercialization plans for their first instrument. So these are just to say that we finished up the planning process and got the RFA out. So my acknowledgement slide is for the people who are developing the technologies. I've run a little bit long, and I do apologize for that. But I hope you'll be able to stay for a few questions if you have. Thank you.
We do have time for questions. Since it's a little hard to hear in this room, when you're asking a question, if you would stand and if you would then try and speak up.
This is very interesting. I understand that at dinner you talked about the X chromosome. I believe the X chromosome is the key to using genetic research in genealogy, because the X chromosome is the longest living piece of DNA that's identified as a single person. You have one X chromosome, the woman has 2. One of those is an exact copy of her father's X chromosome. And the only meiosis that occurs in the X chromosome is in the woman 6 weeks after she's conceived. This allows the research to extend back hundreds of years as compared to the other chromosomes. One interesting thing I found was that if you plot the ancestors you inherit your X chromosome from on a piece of paper, according to the fractal pattern, the dimension of that fractal is exactly the golden mean. I think that's unusual. and is part of the reason why it's a good candidate for use in genetic studies for genealogy. Have you found other instances of structures in the field that are fractals and have different fractal dimensions?
That's a very interesting question. I've never heard anyone look for that. It would be interesting to find out. The X chromosome certainly has a number of important genes on it that are responsible for Fragile X syndrome, for example, mental deficiency syndromes. So there are lots of reasons to study the X. Of course, many of the diseases that we're interested in studying are encoded by genes that are on other chromosomes. So for genealogy, X may be the best candidate. For a lot of what we need to study here, though, we do need to study the autosomes and the Y. Thanks.
Thank you.
Yes?
I hesitate to suggest this, but could this technology have something of the dark side? In that if you can take...
Doesn't every technology?
Yes. If you could take a person very quickly, assess their particular DNA down to what makes them essentially unique, couldn't you develop poisons on that person that are selected for them alone and other nefarious little things?
So, I mean, one of the downsides that's a pretty big investment, probably. There are probably easier ways.
Bringing the cost of all of the damage.
There are probably, well, I mean, you could do the sequencing, but then developing the drug that would affect them and only them. That's trickier. There are probably easier ways to off somebody. But on the other hand, the idea of the biological foundation of race and ethnicity. I mean, there are polymorphic markers that are found more commonly in people of some geographic origins than in people of other geographic origins. And so there is the potential to use that to identify people that you don't like that group. And governments, there's been concern that governments could do that. There's the potential to say, well, this whole group of people have a propensity to have sensitivity to this particular chemical. So we're not going to hire any of them in any of our facilities where we have that particular chemical. There are all kinds of ways that we could use genomic information for discrimination. And so we do need some laws, and we're starting along that path to try to prevent those kinds of discrimination. On the other hand, so yeah, as you know, every technology has its good points and bad points. If until we can know for sure which precise variants are dramatically increasing the chance of a particular individual getting particular diseases, there are going to be ways to classify groups of people who have a better chance of being treated with a particular therapeutic approach than other groups of people. And this will help for the drug development process and will help with prescribing those therapeutics. So, there are lots of landmines in any new technology. One of the things, I have some other ideas that I can share with you, but that are kind of related to that, but let's get to the question. Yes?
When you do a genome, isn't there a lot of so-called this junk DNA that's out there? And when you do that, do you do the complete thing, including the junk? And is there a way now of analyzing that to see there's supposed to be some kind of potential catalytic effect of the so-called junk?
Yeah. So that opens up another long discussion, but I'll keep it short. So about roughly 5% of the genome codes for proteins. We understand that portion of the genome a lot better than we do the other 95% of the genome. So some people who are, I had the word exome on one of the slides, and I didn't stop to explain it. Some people, instead of sequencing whole genomes, are sequencing just the 5% of the coding regions, because it's much easier to interpret. And some of the simpler, if you will, diseases, particularly like the Mendelian diseases, are where if you have that change in your genome, you have that disease. It's often because it interrupts a protein coding region. And it either changes the protein so it doesn't function correctly, or it terminates, it shortens the protein, truncates the protein, so you don't have it, essentially. So those are much easier to interpret when you find a change in those genes. However, what people are finding, once we have the tools to look at other regions, is that there are changes outside of the protein coding regions, in the regions that turn the protein coding regions on and off, or up and down, that modulate their expression, that appear to be responsible, very strong evidence, appear to be responsible for some of the complex diseases, or contributing to some of the complex diseases, diabetes, heart disease, the things that really affect very large numbers of people. So as we have new tools, and essentially can shine light on things that we couldn't look at before, we're discovering that there's a lot of important stuff in that part of the DNA that some people used to call junk DNA. You know why it's called junk, right? And junk instead of garbage. Junk is the stuff you keep around, just in case you need it, as opposed to garbage which you throw away. No, and I think that's proving to be true. There's stuff going on there, we just didn't understand it before.
I'll give Wikipedia as my source for this in case anybody wants to check it out. But and perhaps you'll run across this fact that there are extremely strong reasons to believe that humans are more closely related to orangutans than they are to chimpanzees. All right. That's a very strong argument for that. And yet, when you do the DNA analysis, humans are more closely related to chimpanzees. So what does that tell you about using DNA analysis?
I think we need to look at the data and how people drew those conclusions. The relationship to orangutans is based on what?
Well, I'm going to summarize it. I just suggest.
Yeah, I mean, I don't know the basis for the claim in Wikipedia, which I understand you're quoting.
Well, they just repeat the facts, but okay.
Yeah, I wondered for a while if there's a trend toward concentrating gene therapy on the transcriptome, since, as you say, it basically is the end result of all of the gene splicing and so on. It seems like a simpler way to approach remedying problems.
Yeah, I mean, well, there are lots of challenges to gene therapy. There may be other ways if what you're trying to do is change the regulation of a gene, rather other than doing gene therapy. But if you actually have to change the structure of a gene because it produces a protein or a structural RNA, that if that structure isn't right, there are fewer ways to correct that problem. So I mean, I think the way you're thinking about it is probably right. If you're going to go to the trouble of gene therapy, it's probably going to be used mostly for correcting structural defects in a protein or a structural RNA. I think that's probably right.
One more question?
If you would, I was just hoping, with the love of gadgets in mind, you had briefly talked about the quality of the readings, and you had a chart up there with all the different types of equipment and their different methods listed. And I was wondering, how are you quantifying the quality of the readings, and what are the correlations and evaluations of the different types of machinery?
Yeah, excellent question. So, of course, if you don't have the truth, it's really hard to test quality. So we do it in a number of different ways, and I'll just describe a couple of them. When people sequence with different machines and look at the variants that they've called based on the two different technologies, they find that something like 70 or 80% of the variants that they call are the same with the two instruments. But it's a Venn diagram, and often the sides of the Venn diagram is the stuff that you only see with one technology and the stuff that you only see with the other technology. So, and then you go in and do laborious tests to find out which of those things are true. And you find out that stuff that's over here and stuff that's over here is real when you do, as I say, laborious biochemical tests. So they're really in those genomes. It's just that one technology missed it altogether. So you can sort of trust the stuff that multiple technologies are seeing, and maybe it's actually true. And then you have to use other, as I said, more complicated, laborious, expensive tests to look at that other stuff. But we also have these other technologies. So we have microarrays, and a lot of the microarray technologies have been tested over and over and over again. And so and there's pretty good, they have pretty good validation. So a lot of the studies that are done with the new technologies validate against SNP data, validate against microarray data. So we do have, basically you compare the results from different sequencing technologies, but also with sort of orthogonal technologies that get at the data in different ways. There are lots of individual test tube tests that you can run on individual genes. It's challenging to run them for the whole genome, but there are ways to do those kinds of tests to validate. So those are some of the ways that people are going about this. But it's a real problem.
Thank you very much.